All right, so chapter 35 is an introduction to the immune system. So we'll go over some vocab, we'll go over some pathogens, and we'll talk about innate immunity and acquired immunity. So a pathogen is anything that causes disease, and I think a lot of times people think about like human pathogens, but pathogens can infect a variety of animals, and you can have pathogens that infect protists and bacteria and plants as well, but since this is the animal unit, we're going to focus on animal immune systems. So pathogens that infect animals include bacteria. Their major entry point is through direct bodily contact, like open wounds, inhalation, and also ingestion. And then you've got viruses. Viruses, at the most simple form, have a nucleic acid and a protein coat. Some of them have an envelope as well. And viruses have to take over their host cell's machinery in order to replicate themselves, and sometimes they kill host cells rapidly, or sometimes they lie dormant for a period of time and then ultimately kill the host cell. And some viruses can insert themselves into the host cell's genome and interfere with the cell cycle and even cause cancer. Then we've also got eukaryotic parasites as well. So protists can be parasites, fungi can be parasites, or different worm species can be parasites as well. And typically, parasites are going to damage the host by using the host's nutrients or secreting toxic chemicals that the host has a response to. So with immunity and immune systems, immunity is the ability of an animal to ward off internal threats like microbes or microbial toxins or cancerous cells, basically anything your body can recognize as foreign. And then the immune system is actually the cells and organs that help contribute to immunity. All animals have an innate immunity, which is the defense that is active immediately upon infection, and then vertebrates also have an adaptive immunity, which has to be educated in a way, and we'll talk more about that coming up. So this is not from our book, but I really like this picture. It shows you the innate defenses and the acquired defenses, and then it breaks down your innate defenses, which again are present at birth or not specific, into big categories. Really, your first line of defense is your skin and mucous membrane, so your surface barriers, and then you have non-specific internal defenses as well, like phagocytic cells. Remember, phagocytic cells are cells that eat things. Natural killer cells. You've also got inflammation and antimicrobial proteins, and we'll hit those up coming up here in a second. And then you've also got your acquired defenses, which maybe if you've taken another class, they've called it your adaptive defenses or your specific defenses, and these develop after exposure, and this includes your humoral immunity, which is your B cells, and your cellular immunity, which is your T cells. So again, innate immunity is not specific. It doesn't require prior exposure for the innate immunity to work. It recognizes kind of a general conserved property that says this is not supposed to be in your body. This is something that is not me. This is something foreign. And it helps to protect against foreign cells or substances without having to tell specifically what they are. Innate immunity is inherited to some degree, and this includes your body surfaces, your phagocytic cells, and your natural killer cells, inflammation, and antimicrobial proteins. And we're going to knock through each of these coming up. So I think body surfaces is pretty straightforward, right? It's your first line of defense. Not very many microorganisms can actually penetrate intact body surfaces. And then to help with that, you also have glands that secrete antimicrobial molecules like mild acids, enzymes that are hydrolytic and break molecules down. And then you also have mucus, which is a sticky trap to help stop invaders, and mucus is also antimicrobial. And then you have hairs that help prevent pathogens from entering in your body, and then stomach acid also can help denature any proteins of any pathogens. So your body surfaces are really your first line of defense, and that's part of your innate immunity. You also have innate immunity cells, like phagocytic cells that engulf particles and destroy them. And you've also got mast cells, which release histamine, which is involved in the inflammation, which we'll talk about in the next slide. You've got basophils that secrete heparin and histamines, and natural killer cells. They do just like what they sound like they're going to do. They recognize things that are foreign and then they kill them. Or they also recognize general properties of virus infected cells and kill them as well. Because if we can kill the cell that has the virus in it, then we can help stop the virus from replicating. So then with inflammation, I think people think of inflammation in a bad way, but some amount of inflammation is helpful. A mild amount of inflammation is not a bad thing. So inflammation is a localized response to infection or injury, and what it does is it increases blood flow to the area. So if you have more blood flow, you have more white blood cells that can attack any pathogens and clean up debris. You also have more proteins, which can either be antimicrobial proteins or part of the repair process. Inflammation also increases vascular permeability, which again allows these things to exit. And then another important process of inflammation is your phagocytic cells and your mast cells. So inflammation helps to destroy or inactivate any foreign substances or pathogens, and it helps clear the area of dead cells and really sets the stage for tissue repair. So mild inflammation is helpful. This is not the same as like chronic inflammation, which can be damaging. So then antimicrobial proteins include interferon and complement, and often people don't pay attention to these. Like the book has like maybe one paragraph on this and it's teeny tiny, but I think these are really interesting and I think it's worth talking about. So interferons inhibit viral replication inside of host cells, and they're not specific to a particular virus. And then complement system is called complement because it complements antibody action, and we'll talk about antibodies coming up here, but complement basically kills microbes without phagocytosis, so a cell doesn't have to take the microbe in. Instead, complement system uses something called the membrane attack complex, which creates basically a pore or a hole in the bacterium, and then it causes fluid to rush into that bacterium, and that causes that bacterium to burst. So this picture is showing you interferons. So basically what happens here is the virus is going to enter a cell and uncoat and start making copies of itself, but that also switches on an interferon gene. And so the cell, it's still going to produce virus particles, but the cell also produces interferons. So you can see the little triangles there at the bottom of the screen are the interferons. And so the first host cell releases the interferons, and then the second host cell gets a message. So the interferon is like, hey, there's a virus. We really need to not let that happen. So it warns the second cell that the virus is coming. So the interferon tells the second cell, okay, you need to make some antiviral proteins. The second cell makes antiviral proteins, and those proteins block viral replication or reproduction. So then back to complement. Complement proteins cascade and they create this hole in the cell called the membrane attack complex. So complement proteins are going to form this pore, and then that pore will allow fluid to rush into the cell, and that will cause the cell to lyse and burst. So which antimicrobial protein triggers cells to produce antiviral proteins? Is it complement or interferon? So complement complements antibody activity, and complement is what pokes a hole in the cell. Interferon interferes with viral replication by producing antiviral proteins. So one is B. And then what is the first line of defense against pathogens? Your skin and mucous membranes. So your surface barriers are your first line of defense against pathogens. If you said innate immunity, like I would take that, but specifically on the innate immunity, I'm looking for skin and mucous membranes or barriers. So we talked about how our innate immune system can detect and destroy or prevent pathogens from spreading, but pathogens have also evolved to evade the innate immune system. So some pathogens can avoid destruction by phagocytic cells. Some pathogens have capsules that interfere with molecular recognition, and some pathogens even resist breakdown within lysosomes after they've been engulfed by a host cell. So there's a number of ways that pathogens get past innate immunity. So that brings us to acquired immunity. So your acquired immunity is specific, and it's found in all classes of vertebrates except for jawless fish. Remember your cyclostomes. And acquired immunity is going to require exposure to the foreign substance. The foreign substance is called an antigen. So any molecule that your body doesn't recognize as itself is called an antigen. And it's called an antigen because it's an antibody generating molecule. So sometimes people get stuck with antigen versus antibody, and it's definitely worth spending a little bit of time on it because an antibody attacks an antigen. So the antigen is the foreign thing, the antibody is the thing your body makes in response to the antigen. So be careful on that one. As far as your acquired immunity goes, there's two kind of wings of this. You've got humoral immunity, which produces your antibodies and is with your B cells. And then you have cell-mediated immunity, which is largely a function of T cells. So I know we already talked about the lymphatic system a little bit last chapter, but the lymphatic system is important in these specific defenses because you've got lymphocytes that are going to circulate in the blood and also lymphatic system. And you've got lymphatic vessels that are going to collect the lymph and return the fluid to the blood plasma. And you've got these lymphoid organs that are going to help differentiate the lymphocytes and help them become mature immune cells and supply these to the lymphoid organs where they're going to multiply. So the lymphocytes are going to circulate through the lymphoid organs, the interstitial fluid, which remember again, that's just the fancy word for extracellular fluid, and then back to the lymphatic system again. And this increases or increases the chance that they'll run into an antigen and then be able to respond to that. So here's just kind of a picture with your different lymphoid organs. So like you've got the thymus and the bone marrow. Your T lymphocytes are going to mature in the thymus, your B lymphocytes are going to mature in the bone marrow, and then you've got things like the tonsils and the lymph nodes and the spleen, and you've got regions in the small intestines and your lymphatic vessels as well that are part of the lymphatic system. And then if we kind of zoom in over here on the right hand side of the picture with a lymph node, we talked about this last chapter, but notice that there's more lymphatic vessels going in than leaving the lymph node. So this gives the lymph node time to process the lymph and kind of educate these lymphocytes and gives them time to break anything down. So again, with lymphocytes, B cells mature in the bone marrow, and some of these form plasma cells that are going to produce antibodies, and this is part of humoral immunity. And then you've got T cells or T lymphocytes, and those mature in the thymus gland. Are you seeing a pattern? B cells, bone marrow, T cells, thymus. So T cells include cytotoxic T cells. If we look at the word cytotoxic, cyto means cell, right? So these are cells that attack other cells. So cytotoxic T cells directly kill infected or mutated or even transplanted cells. If you receive a transplant, oftentimes you need to be on immunosuppressants so that your own immune system doesn't attack those transplanted cells. Then we've got helper T cells, which again do just like what they say they're going to do. They help activate cytotoxic T cells and they help activate B cells. So they're real good helpers. And T cells are part of cell-mediated immunity. So both humoral immunity and cell-mediated immunity are specific. And then natural killer cells are similar to T cells, but they're really kind of more in that non-specific or in the innate immunity. So in order for the acquired immune system to respond to an antigen, first it has to recognize that antigen. So during development, each lymphocyte is going to have a particular membrane receptor, and that receptor is going to bind to a specific antigen. So like one particular lymphocyte can recognize one particular antigen, and then that's going to activate the lymphocytes. And so any lymphocyte that has bound to an antigen, it's needed because the antigen that matches it is in the body. And so that lymphocyte's like, oh no, there's trouble. We better make a bunch of copy of ourselves and deal with that. So they make lots of clones. And then to actually attack the antigen, B cells are going to differentiate or mature into plasma cells and secrete antibodies. We'll talk about what the antibodies do coming up, but basically they're going to recruit and guide other molecules and cells to the area. Then cytotoxic T cells themselves are going to directly attack and kill the antigen-bearing cells or the antigens themselves. And then plasma cells and cytotoxic T cells eventually will have to die by apoptosis to prevent an excessive immune response. However, if we run into the same antigen again in the future, you have memory cells that are going to persist and be ready to attack that pathogen the next time. So here's just kind of a picture that shows you how both humoral immunity and cell-mediated immunity are going to respond to the presence of an antigen and how they really kind of work together and kind of overlap. So you've got the antigen, and that's going to be recognized by B cell, helper T cell, and a cytotoxic T cell. So helper T helps activate B cells to mature into plasma cells, which then secrete antibodies. Helper T cells also help cytotoxic T cells mature and make sure that they don't attack your body cells. And then in order to stop the attack, once we've dealt with that antigen, we have memory B cells and memory T cells. So antibodies come from mature B cells. The B cells mature into plasma cells, and then the plasma cells release antibodies. And those B cells have to be activated by both an antigen and helper T cell. So antibodies inactivate antigens. They help destroy antigens, and they do this by neutralization, agglutination, precipitation, and complement activation. We'll talk about that with pictures on the next slide. And then as far as antibodies go, you maybe heard of immunoglobulin or the Ig protein family. Like mammals have 5. We've got IgM, IgG, IgE, IgA, and IgD. All vertebrates have IgM, and they have different roles, but we're going to keep on going because we just kind of summarize things here. So don't worry about remembering the IgM, IgG, IgE, IgA, IgD. But if you take microbiology, I think you'll go over that in more detail there. So as far as what antibodies do, antibodies bind to antigens, and they inactivate them by neutralizing them. So they block viral binding sites, they coat bacterial toxin, so that neutralizes them. Agglutination is like the $5 word for clumping. So antibodies help clump up bacteria, or they can cause precipitation of dissolved antigens. If you've taken chemistry, that's the kind of precipitation we mean. Anyhow, neutralization and agglutination and precipitation all enhance phagocytosis. So I found that picture there of the like angry looking white blood cell, or the very hungry looking white blood cell in the right hand corner, and then the sad looking microbe with the little Y-shaped things on it. Those little Y-shaped things are the antibodies. Another thing antibodies do is they activate the complement system. Remember, complement is an antimicrobial protein, so the complement system, it takes a cascade for them all to arrange in this pore forming complex, and the cascade takes less steps if antibodies are present. So they help activate complement system. And then on the bottom left hand side there's a picture of an antibody, and it shows you that they have different binding sites for different antigens. So after the first time you've been exposed to a antigen or a pathogen, you get immunological memory if your B and T cells responded to it. So the primary immune response is your first exposure to the antigen, and your antibody production is going to be slower and at a lower level. The secondary immune response happens when you've been re-exposed to the same antigen. So your body recognizes this, and so the immune response is going to be faster because you already have those clones, those memory cells. And you'll have larger antibody production. So your memory B cells are going to be quickly stimulated to differentiate and multiply. And again, you have this immunological memory. So if we look at this graph, this is showing you the primary response and the secondary response. On the y-axis of the graph, you've got antibody titer, so like the antibody concentration, and then down on the x-axis, you've got the time there in days. So the first exposure to antigen A happens at day 0, and you can see, it takes a little over a week for you to get your peak antibody production there. And then it goes down around, 28 days after primary exposure. Then if you were exposed at day 28 to antigen A and then also to antigen B, your antigen A response would occur much more quickly, and you would have much higher concentration or production of antibodies, but your antibodies to antigen B would be just like the first time you were exposed to antigen A, because this is the first time you're exposed to antigen B. So that's basically showing you that your secondary immune response, you get more antibody production, and it's faster, and they last for a longer period of time. So when we talk about humoral immunity, we can kind of break that down further as well. You can either have active or passive humoral immunity, and then your humoral immunity can either be naturally acquired or artificially acquired. So active humoral immunity is when you, or whatever organism we're talking about, when you or their B cells encounter specific antigens and produce specific antibodies against those antigens. So humoral immunity can either be naturally acquired or artificially acquired. If it's naturally acquired, that could be like through response to bacterial or viral infection. If it's artificially acquired, your B cells are still going to be doing the work, but they could come across a vaccine of a dead or weakened pathogen and still respond to that. So as long as you're making it or the organism is making it, then that's active humoral immunity. Passive humoral immunity is when you get antibodies from another source. So in this case, your B cells would not be challenged, so you have no immunological memory here. So naturally acquired would be when antibodies cross the placenta to an infant, or through milk, could be another form of naturally acquired passive humoral immunity. Artificially acquired humoral immunity is when you get injection of a serum that contains the antibodies. Those antibodies were made in another organism. Oftentimes like horses are involved with this. Either way, you didn't make them and your mom didn't make them. You got them in an injection there. So you're still not going to have a memory. So that brings us to cell-mediated immunity, which is the role of T cells. The T cells have different receptors for different antigens, just like B cells. However, unlike B cells, the T cells receptors are going to remain embedded in their cell membrane. They're not going to be secreted like antibodies from B cells. So just like B cells, there's multiple DNA rearrangements made during T cell maturation that allows for different receptors to be produced in the mature T cells. And T cells can't combine with an antigen unless it's complex with the majohistocompatibility complex 1 receptor, or MHC1. So don't get too hung up on the MHCs right now, but basically MHCs are kind of like cellular identity tags or genetic markers of self. There's class 1 MHCs that are found on all of your body cells except for your red blood cells or erythrocytes, and a CD8 protein on cytotoxic T cells is going to bind to class 1 MHCs and trigger apoptosis, and it releases perforin, which perforates these cells and triggers them to be destroyed. But right, what's the role of cytotoxic T? Destroy cells. So there you go. Class 2 MHCs are found only on antigen presenting cells, which are sometimes called APCs, also macrophages, B cells, and dendritic cells. And class 2 MHCs are going to bind to CD4 on helper T cells, and that helps activate the T cells and B cells. It induces T and B cell proliferation or cloning, and it helps to activate macrophages. Macrophages are the phagocytic cell. So if we look at this diagram, there's kind of a lot going on here, but number 1 starts up on the top left-hand corner here, where we've got a bacterium entering in through a cut in our skin surface. That bacterium is going to be carried into the lymph node, which they've got this big green blob here is your lymph node. And then a macrophage right here takes that in, and the antigen is going to be processed and then presented to a helper T cell here. So see that little yellow dot? It's presented like, hey, helper T, check this out. Helper T is activated and then divides and also secretes molecules to activate B cells. And those B cells mature into plasma cells. And remember, those plasma cells are going to secrete antibodies that are going to inactivate antigens. So which type of cells secrete antibodies? Hopefully you said plasma cells, maybe you said B cells. Honestly, I would take either of those, but plasma cells are mature B cells. What happens when antibodies bind to antigens? So antibodies inactivate antigens. They can do agglutination, precipitation, neutralization. Those things enhance phagocytosis, or they can activate complement system, which forms that membrane attack complex, right? And that pokes a hole in the cell and causes the cell to lyse. And what's the difference between TC and TH cells? Well, TC is cytotoxic T. TC is cytotoxic T. That's so hard to say. TC is cytotoxic T and TH is helper T. So cytotoxic T cells are going to directly kill cells, and helper T cells help activate cytotoxic T and B cells. So we get different B and T cells by combining different sections of DNA. So you combine things called variable elements, and that can give you lots of different receptors, like millions of different receptors. And the capacity to generate this diversity is built into the Ig genes, and many different genes can be produced from the same gene by swapping around the DNA. And this swapped around DNA is going to be transcribed, so go from DNA to RNA, and then translated, so go from RNA to protein, and you get different antigen receptors formed. So you can have a receptor light chain gene that contains a variable segment and a joining segment and a constant segment. Obviously the constant segment is going to be the same, but then the gene has this one same constant segment and then 40 different V segments and 5 different J segments. So you can kind of shuffle the V and J segments around and get a whole bunch of different kinds of receptors that way. So up here, you've got the DNA of an undifferentiated B cell, and you can see that there's the one C segment here. There's different J segments and then different V segments. And then when those are going to be combined, so you're going to have recombination that deletes DNA between the V segments and the J segments, you get this functional gene down here, and this part of the gene is going to be transcribed into pre-mRNA and then edited into mature mRNA, and then you get the protein produced from the mRNA, and you can see how you can have different antigen receptors with different V segments there. So ideally, your body would not attack itself, although sometimes, unfortunately, that does happen. But what's supposed to happen is that when the lymphocytes mature in either the bone marrow if they're B lymphocytes or the thymus if they're T lymphocytes, they should be tested for self-reactivity. And then any B or T cell that has receptors that are specific for the body's own molecules are going to be destroyed. And so that way they just won't be used and your body won't attack itself. Obviously, sometimes that doesn't occur, but typically it should and does. So we talked about different types of pathogens. So recall different types of pathogens. We had virus, bacteria, and then parasites like parasitic worms or even protozoan parasites. Then for innate immunity, remember that doesn't have to be exposed to something first. It's just innate. We're born with it. So you've got your skin and mucous membranes for barriers. You've got your cells like your NK cells and your phagocytic cells. And then you've got antimicrobial proteins like complement system and interferon. Also, inflammation should be up there. I'm not sure why I didn't put it there, but inflammation should be there as well. With acquired immunity, you've got your specific defenses here. So your humoral side is your B cells, which mature to plasma cells and release antibodies. And then you've got the cellular immunity, which includes cytotoxic T cells and helper T cells. And then under humoral immunity, notice I have active and passive immunity. Remember, active and passive is whether or not you make the antibodies. So if it's active, you made antibodies. If it's passive, somebody else made them for you, like your mom. Thanks, Mom. And then both active and passive can either be natural or artificially acquired. If it's natural and active, you made antibodies in response to an infection or a pathogen that you acquired. And if it's passive humoral immunity that's naturally acquired, you got antibodies through the placenta or through milk from your mother. And then if it's artificial, if it's active and artificial, that would be from an injection of dead or weakened antigen, but you still made the antibodies to that. If it's passive artificial, that would be an injection of antibodies that were created in another organism. So that's kind of the overview of immune system there. As always, feel free to reach out if you have any questions. I'm happy to help you out with that.